So we are trying to finish up our discussion of the major structural parts of the brain. We talked about the brain stem, talked about just the brain stem. That's it. We are on to the cerebellum. We're looking at the Diencephalon today, and we'll discuss the cortex. We will probably not be able to get into the functional areas, but we'll talk about the structures, some of their roles, but the functional breakdown we'll leave for next class. So the cerebellum, the mini brain, it occupies the inferior and posterior aspects of the cranial cavity. The cerebellum is this little wedge-shaped surface. It's got a lot of really small, fine ridges, a lot of folds. It has two hemispheres like the larger brain does, the larger cerebrum. We'll get to that in a second. And this defining characteristic down the midline called the vermis, which loosely translates as worm. I guess it looks like a worm. And the cerebellum is critically important in governing and executing movement plans. So when you plan to get that can of soup out of the cupboard, or you plan to go for a run after class, or you're going camping, you load the car up, whatever the movement is that you need to do, the cerebellum is critically important in governing the execution of that plan. It takes the voluntary signals, it sends them to the periphery. The plan, the efficient plan, is largely created and stored in the cerebellum. This is a dense area of the brain. Many folds. It might be about 1/10 of the size of the entire brain, but it contains half of the nerves. So it is densely packed. Remember, the cerebellum governs the movement plans, but it can be modified. We talked about last class how it receives input from the olives in the medulla. If a plan isn't working exactly right, if you keep falling off of your bike, it receives that afferent input, that sensory input, to adjust the movement plan. And then hopefully over time, that plan becomes such that you don't fall off your bike and you can ride in a straight line indefinitely. So it governs the execution of efficient movement plans in the body. Let's take a more in-depth look at how it relates with the other areas in the brain. So we're looking at a superior view here. This is an inferior view. It looks pretty much the same except for this white part. And the white part are the cerebellar peduncles, which is a fun word, which translates as little feet. They look like little feet, and they are the points where the cerebellum connects to the brainstem. This is the point of attachment where neurons can arrive and leave the cerebellum and head out to the rest of the body or other areas in the brain. And they're white on this diagram because they are white matter. And this is one of the first times that we've really talked about white matter versus its counterpart gray matter. You might have heard about them before, but white matter tracts. Remember that word from last class? The conduits for nerves to send signals through the body. White matter tracts are generally conduits where signals are relayed or transmitted. And the counterpart is gray matter that contains the cell bodies, the business end of the neurons. That's where they connect to other neurons, where the cell bodies lie, where the processing, the integration, the decision-making, the calculations, where those higher order or more complex actions occur. So white matter, typically it's located inside, deep to the surface, is mostly a conduit where signals are sent, and the outer layers, also referred to as the cortex, and we'll see that coming up again today. The gray matter contains the bodies of the neurons that connect to other neurons where synapses and calculations occur. So the connection point are white matter tracts that connect the cerebellum to the brainstem. And you can kind of understand what they look like here, but an easier or maybe a more intuitive way to look at it is to slice the cerebellum in half down the midline. Let's look at a sagittal plane from the right looking towards the left. You can see the white matter deep to the surface. The gray matter are these reddish brown staining circular areas towards the surface superficially. You can see the difference obviously in why they're called gray matter versus white matter. The pons and the medulla for reference. And this structure is referred to affectionately as the arbor vitae because it looks like a tree as it spreads out into the cerebellum. You can kind of think of the white matter versus gray matter as a bouquet of flowers, right? The flowers will fan out on top. They take up a lot of space, and those are the bodies of the cells. You can fit more flowers together if you lift some up or push some down. You give them different levels to sit at. And that's what happens a lot of the time at the surface. There's a lot of these folds to fit more in. And then the stems of the flowers come together. They converge. The stalks are the gray matter that transfer nutrients and water, etc. So it's somewhat of an analogy that you can use. But back to the peduncles, so the little feet, which are these surface structures that are the endpoints, the termini of the white matter within the cerebellum. There are three regions of interest that we're concerned with. And in understanding that the cerebellum helps organize efficient movement plans, it should be no surprise to see where the nerves in these general areas connect. So the superior region connects to the red nuclei in the midbrain. And we talked about those last class. The red nuclei and the substantia nigra are critical regions in unconscious movements. When they're damaged, the symptoms of Parkinson's present. And a linear motion reaching for the can of soup turns into something a bit more convoluted. And so that information arrives at the cerebellum through the superior peduncles. The middle peduncles relay information through the pons. So this is where the brain as a whole, the cerebrum, decides to do an action. You voluntarily start walking out of class because you're bored. The signal to contract those muscles and start walking is relayed through the pons, through the middle peduncles to the cerebellum, and then onwards to the musculature of the legs. So the voluntary or the somatic nerves, that functional branch of the nervous system, arrives at the cerebellum through the middle peduncles. And then the inferior portion is a little more varied, a little more general. We have a number of different afferent signals or sensory signals arriving. Some from the inner ear, which is really important for maintaining balance. I don't have to consciously think about maintaining my balance. I've practiced over a number of years. And that information is constantly being relayed to my cerebellum if I adjust through these inferior peduncles. This is also where that modifying information arrives. The modifying information from the olives of the medulla. If I happen to fall over, whatever my plan was for maintaining my balance didn't work. And so signals would arrive at this location to modify that plan in future. Hopefully I wouldn't fall over as frequently in future and I could maintain my balance. Or hopefully I learn how to ride a bike better or slackline or whatever you're into nowadays. This is how you learn and modify that plan. So critically important area for the control and organization of unconscious skeletal muscle movements. Cerebellum. Make sense? Okay, let's move on. Note, these are high-level overviews. There are a lot more details, but for our purposes as human kinetics students, this is okay. We have a good foundational understanding of what these areas are supposed to do. We can move on from there. The Diencephalon, apologies, immediately superior to the midbrain, right? Sits on top of the brainstem, articulates with the midbrain. Cerebellum was behind those two structures. The Diencephalon has three really important areas, and I want to set the stage here. The Diencephalon you can see highlighted in green. Notice that it tends to surround the third ventricle. We looked at the ventricular system where cerebrospinal fluid is produced from the lateral ventricles. It combines in the third ventricle in the middle, moves to the fourth, and then moves out around the brain and the spinal cord. And by the way, I put up an image on Moodle just to show you the apertures that I couldn't get working with the app yesterday, so check that out. It's in this section on Moodle. These structures surround the third ventricle. The opening with the cerebrospinal fluid is inside of the Diencephalon, and that becomes important in a second because we monitor the cerebrospinal fluid through some of these structures. So the thalamus, the hypothalamus, and the epithalamus, often thought of as the primitive brain, so to speak. The thalamus is the big, roundish, olive-shaped, two-hemisphered mini brain, the obvious structure that sits just superior inside or around the third ventricle. This is one of the relay stations where all the sensory information, except for smell, passes through to be raised to the cortex. Passes through to be raised to the cortex. So remember when we talked about the reticular formation yesterday? And the reticular activating system, the tracts, the conduits where the signals are sent, where you become aware of sensory information. A lot of that information, you'll notice, passes through this dark brown staining circle, which is the thalamus. It's a relay point for a lot of that sensory information, for you to become aware of things that you need to become aware of and ignore things that you don't. So just looping that back in so you understand how they fit together. I'm not going to ask you about all the slices and colors and names of the thalamus. What I want you to understand, this is a complex diagram and it looks really busy, but the colors here correspond to the larger cerebrum. And we're not going to talk about the cerebrum yet, but I'm bringing this slide up here because the colors correspond. The matching colors are where those signals travel from the thalamus to the higher order regions in the brain. So you can see the red area on the left-hand side of the thalamus sends nerves to this dark staining red area on top of the cerebrum. And so we might be able to tell what nerves in this area do if we understand what this area of the brain is trying to control or what it perceives. And we will later. But I just want you to know that the colors match up. It indicates where the nerves travel from in the thalamus to in the larger brain areas. I also want to take a moment. What's not shown on this slide is that on the underside, the ventral side of the thalamus, the limbic system, or the emotional brain, makes connections, which could be why some senses trigger memories or emotions. Sight, touch, taste might relate to emotions or some memories that you've had in the past. And we won't go into detail on this. I had a few slides later on, but in the interest of time, I'm not going to go into those in detail. I just want you to know that there are connections between the system in the brain that specifically deals with emotions and then sensory information, sight, touch, taste, hearing. Smell is different, that comes out later. So the limbic system, or the emotional brain, makes connections on the ventral surface of the thalamus. What I find really interesting in this area, given my area of research, is the hypothalamus. The hypothalamus, under the thalamus. The name tells you where it sits in the brain, under the thalamus. You can see the thalamus, that's the rounded projection here, on top, on the side, pushing into the third ventricle. This big open space is the third ventricle. And all of these colored blobs are parts of the hypothalamus. And what you'll want to notice right off the bat is that these bodies, these nuclei, the colored parts of the hypothalamus, line the wall of the ventricle. That will become important. They're in the wall of the ventricle, and so they're washed over, they're bathed in cerebrospinal fluid. All of these 4 major regions and 12 different nuclei. I'm not going to ask you the names of each one or even the divisions of the 4 major regions. I just want you to know what I want you to know. A lot of stuff written here is what I want you to know. What I want you to know is the hypothalamus is critical in hormonal regulation of homeostasis. Critical in regulating blood pressure. There are centers for respiration. There are centers for regulating fluid balance. The hypothalamus is sensitive to changes in homeostasis and works really hard to maintain homeostasis. Nothing happened. I wonder what area of the brain is responsible for reflexes. The hypothalamus, tightly involved in maintaining homeostasis. It does that through direct nervous stimulation. So these areas of the brain send nerves to organs that will respond to a disturbance. Or you'll notice that it's connected to this dangling apparatus, immediately anterior and inferior, called the pituitary gland. And this is a gland you've probably heard of before. It's considered the master gland in the body, regulating growth and development, secreting important hormones, which we'll talk about at the end of this semester. Release of those hormones is controlled by the hypothalamus. So it has powerful ability to regulate homeostasis in the body through the connection to the pituitary gland through the infundibulum, which is a fun word. This is where the missing sensory information gets relayed. The sense of smell or olfaction that wasn't included in the thalamus gets relayed through the hypothalamus and brought to conscious awareness. So there's some moderate separation. The olfactory senses arrive at the hypothalamus and then are sent to the brain. The rest of those senses are relayed through the thalamus. So it's the odd one out. It's notable. Might be something to make note of. The roles of the hypothalamus are wide and varied, and so this is giving you a sense of some of the sensations and processes that are involved or related to activity of the hypothalamus. Emotions, rage, anger, pleasure, basic sensations like thirst or heat or discomfort, hunger, circadian rhythm is in part regulated by actions of these nerves as well. Many different basic processes regulated by the hypothalamus. So many varied functions, relaying olfactory senses, and the parts of the hypothalamus are in the walls of the ventricle. No more detail than that. The epithalamus. Did you get that last slide? Furious typers. So the epithalamus also contributes to some hormone release. It lies superior and posterior to the thalamus, and the gland that it controls is called the pineal gland. We're not going to spend a lot of time on the pineal gland, but I do want to reference, because we talked about melatonin in class yesterday, and how the reticular formation releases melatonin. It does so through activating the pineal gland. And so melatonin is secreted by this gland. It's regulated by the epithalamus. And there are other important nuclei, which we'll talk about in a second. But melatonin release. Melatonin release. The highlighted area is the epithalamus, the section at the back of the third ventricle. Melatonin release. Some visual signals on their way to being processed. We don't know where that is yet, but it's at the posterior section of the brain. Now you know. On their way to being processed will arrive at the epithalamus and activate or not the pineal gland. What signals will activate the pineal gland? First of all, we want melatonin release to be low during the day, so we stay awake, and high at night, so we fall asleep. And luckily for us, the visual signals that we receive that arrive here corroborate that activity. There's lots of light around us right now. The light is intense, it's bright, and so the visual afferents that arrive at the epithalamus are turning off the pineal gland. I don't want melatonin to be secreted right now. I don't want to fall asleep. Neither do you. You want to stay awake. When the sun goes down and it gets dark out, the light surrounding us is no longer bright. It's no longer abundant. The signal to suppress melatonin release, therefore, goes away. Pineal gland starts to secrete melatonin, but it can be disrupted. And it's very sensitive. The visual signals that arrive at this gland are very sensitive to blue light, which is the light that your smartphones give off, the unfiltered light from your computer screen if you don't have night mode on. They activate those visual afferents. They suppress melatonin release at this gland. It's getting dark. Other hormones are saying it's time to sleep, but the pineal gland is saying, no, I'm seeing light. I'm getting a signal there's lots of light out, so I'm not going to release melatonin yet. The other nuclei, or parts of the epithalamus that I mentioned, the habenular, I always have a hard time saying that word, habenular nuclei, are the missing link for where the olfactory senses connect to the limbic system. So again, the odd sense out. The thalamus linked the other senses to the limbic system. The epithalamus is where olfactory senses are linked to the emotional brain. And it's here that a smell might trigger an emotion or might trigger a memory. The habenular nuclei specifically will relay that sensory information and possibly trigger the emotional brain to have an emotion or a memory. So some notable structures, some notable characteristics of the epithalamus, but that's really all I want to go, or as far as I want to go into detail on that structure. But what I want to circle back to is the fact that these structures surround the third ventricle. And specifically the hypothalamus. You saw those nuclei line the walls of the third ventricle. They are bathed in cerebrospinal fluid. They can sense changes in the chemical composition of cerebrospinal fluid. And that's really important in the regulation of some of these homeostatic processes. The correct name for the areas or the regions that will sense the composition of the cerebrospinal fluid are circumventricular organs. And really that's getting at the fact that these organs are made up of many different parts of the diencephalon. What's important is that they directly monitor changes in cerebrospinal fluid, and they are purposefully exposed. They're not protected by the blood-brain barrier. So things in the blood, things in the cerebrospinal fluid can be directly sensed by these regions. And in sensing different variables in the blood, we can regulate or anticipate the regulation that might be required to keep us alive and maintain homeostasis. So large portions of the hypothalamus make up the CVOs. Some of the epithalamus, the pineal gland and the pituitary gland are also involved in sensing and releasing elements in relation to sensing these parameters in the blood. One example that I like to study, I always study dehydration, sport performance, hockey player performance specifically. When you're dehydrated, your blood concentrates. You're sweating a lot and your blood gets more concentrated, it gets saltier. If your blood gets saltier, the cerebrospinal fluid also tends to get saltier. And the hypothalamus can monitor that. So part of my brain is always monitoring how salty my blood is. And if it gets too salty, unbeknownst to me, it constricts all the vessels in my skin. Why? Well, if I'm sending blood to the skin, I'm sweating. I'm losing fluid from that blood and I'm sweating. This is a protective mechanism to say, you're dehydrated, your blood's getting salty, we can't stand to lose any more water. I'm going to try to clamp down those blood vessels where you might sweat and lose water from in an attempt to keep us alive. If you lose too much fluid, you can actually die. So this is a good thing for staying alive. It's a really bad thing for sports because without blood flow to the skin, you get really, really hot. And when you're really hot and cranky and uncomfortable, you don't want to play hockey. You don't want to go for a run. You don't want to play soccer, go skiing or whatever. Performance really suffers. But physiologically, it's really good that we have these in place. I don't remember why exactly I put this up here. I wanted to show you the pituitary gland. I wanted to point out the third ventricle, and so you can imagine, we've sliced away part of the pons and the midbrain. You can imagine the walls of the third ventricle sandwiched up here underneath the lateral ventricle is where those organs would appear, right in this area. You can also see the apertures. There's a picture on Moodle. The lateral apertures are these little arms projecting out to the side. That's where the CSF will leave the ventricular system. And the median aperture is the little tail at the back. So that's what I was referring to yesterday that we didn't get to see on the overhead. Questions about that? Details, details, details. Let's talk about the cerebrum in the last 10 minutes of class, and we only have like 4 slides. It's not a big deal and a really cool animation. Only 4 slides. Cerebrum. The cerebrum, the last piece in the puzzle, the stereotypical region. When you think brain, you think cerebrum. This is what allows us to develop intelligence and process. It creates the areas of higher order thinking, consciousness, perception, the ability to imagine future states. The cerebrum is the region that allows all of those things, taking in information from the environment, allowing us to put together complex information, synthesize a solution, figure out how we're going to trap that woolly mammoth while escaping from tigers, taking meat with us to survive. The cerebrum is what allows us to do those things. And the cerebrum is a lot like a larger cerebellum. It has an outer cortex made of gray matter and inner white matter tracts, a lot like the cerebellum does. And the functions are very similar. The outer regions are packed with cell bodies. That's where the synapses, the thinking, the processing occurs. And the inner regions are white matter where the signals travel back and forth. When we look at the brain, apologies again, we can see the folds and the grooves, and we give them specific names. The gyri are obvious. The gyri are the ridges, the raised portions. The hills, the top of the folds. Gyri are plural. The singular version is a gyrus. Between the gyri are sulci. Singular version of that is a sulcus. They are the grooves between the hills. And then there are also some really large grooves, which we'll call fissures. Fissures are deep, thin, long folds within the brain. So there's some obvious structural elements that we want to take note of, and they impact how the brain works as well. Those are obvious. Those are part of the cortex. You can see them, and you will in lab next week when you hold a sheep's brain in your hand. When you cut into that sheep's brain, you'll be able to identify the white matter tracts. These are things that relay the signals from where they've been processed in the cortex to wherever they need to go. The tracts will connect different regions of the brain to each other. We'll connect the brain to other parts of the body, and I'll show you those tracts in a really cool animation coming up. Importantly, all the white matter tracts travel seemingly in every direction within the brain, but the two hemispheres, the main halves of the brain, are connected by a really dense knot in the middle called the corpus callosum. The corpus callosum. And I'll show you that in 2 slides. You can't see it from the surface. It's deep to the brain. It's around the diencephalon. Technically, it's just superior to the diencephalon. That's a correction. But it's a really thick white matter tract that connects the two hemispheres of the brain to one another. It's really easy to distinguish in your specimen. So what do these structures look like? You've seen them on paper. I'll show you in a diagram here. If I'm looking from the top, this is a superior view of the brain. I've taken out a small little square. You can see I've cut that square out and moved it over to the left, and we're looking at a frontal section of the brain tissue. You can see the gyri are these raised areas, the hills. One gyrus is shown on the left, and an arbitrary corresponding gyrus on the right-hand side. These green areas are the gyri, or the hills. The sulci, or the dips, the grooves between them, are shown in yellow. You can see a shallow furrow or shallow groove on the left, and a small groove on the right-hand side that separates the gyri. And then the big divisions, the obvious folds, are the fissures. There's one obvious fissure down the middle of the brain, the longitudinal fissure that separates one hemisphere from the other. And if you hold a whole brain in your hand, you can open up the two hemispheres. You can look down the longitudinal fissure. The only things that are holding those two hemispheres together is the corpus callosum that you can look at if you pry those two hemispheres apart. The brain doesn't have these colors. These colors represent different lobes, and generally, lobes will have a certain scope of function. They might deal with motor activities or the functions of certain areas of the body. You can start to see the division of the brain, and we'll talk about the functional divisions later on. But those lobes are shown in a bit more detail. There's 5 main areas of the brain: the frontal lobe at the front, the parietal lobe, which is in the middle towards the side, the temporal lobe, which are at your temples, the sides of your head. If you're looking at this medial view of the brain, this is the right hemisphere, you'll notice it looks kind of like a mitten. The thumb down here is the temporal lobe, and the rest of the hand is everything else. And when you see the brain, the sheep's brain isn't a really good example of this, but a human brain has a really well-defined mitten shape, and that thumb is really obvious. The division between the two is where we house the insula, which I'm not going to talk about right now, and the occipital lobe at the very back, the posterior section of the brain. 4 main regions. The insula is a 5th that we'll talk about eventually, but not right now. Usually, these lobes are colored differently, and they have a scope of different functions. The divisions between them are usually important. So you have the frontal lobe and the parietal lobe, and the line between them is a well-known line called the central sulcus. It's a bit bigger than a small sulcus like you might expect. It's not quite a fissure, it's not very deep, but is an obvious long groove in the brain. And it separates 2 really important regions in the brain. When we talk about the function of the lobes, we're going to refer to in a bit of detail the gyrus anterior and the gyrus posterior to the central sulcus. They have really well-defined roles. I want to sum up today's class with a brief look into the white matter, and then we're done. The outer layer is the cortex, the gray matter where everything is processed. You see all of the undulations which allow you to fit more stuff in. The white matter isn't as obvious. So if we take a sagittal section through the midline of the brain, and then we look at the internal components of the brain, now we start to see the conduits. All these conduits are just wiring, where signals are relayed from different regions within the brain. Now I'll introduce 3 types of conduits and show you a cool animation that you can leave. We have until 5 after. The association tracts connect anterior to posterior regions within the same lobe. So the association tracts run right here above this half-moon-shaped structure. The association tracts go anterior to posterior. Oh, sorry, I had that flip. Wait, no? I'm pretty sure. I think this is wrong. I think that anterior is on the left-hand side. No, it's right. Never mind. Either way, they run anterior to posterior, and that meaty half-moon shape is the corpus callosum. That's that really easy to identify structure that connects the 2 hemispheres together. The commissural tract is what the corpus callosum does. It connects the 2 hemispheres of the brain. It sends signals from one region in one hemisphere to a very similar or the same region in the opposite hemisphere. They're bundled and packed really tightly together because there's a lot of cortex, a lot of neurons that need to pack and send signals through the corpus callosum. We have anterior, posterior, we have medial, we have lateral. The last axis is superficial and inferior. The projection tracts will relay signals from the higher order areas of the brain, the cortex, to the lower order areas of the brain, the brainstem, the diencephalon, cerebellum. Projection tracts, superior to inferior, taking the information that's been processed to the tissues where it needs to go. I'm going to show you on the next slide our most updated, advanced look at what the white matter tracts look like.